So I'll be presenting a brief overview of my recent work looking at connectivity-based personalisation of the TMS target site for the treatment of depression. So TMS is commonly used clinically for individuals with treatment-resistant depression, and these are individuals who fail to respond to first-line behavioural and pharmaceutical therapies. Typically, stimulation is applied to the left dorsolateral prefrontal cortex, as this region has been consistently implicated in depression and is connected to a number of relevant brain regions. TMS is administered daily for around 4 to 6 weeks, and you tend to see this gradual alleviation of symptom severity. Response rates are around 35 to 50%, which is not bad considering this is a treatment-resistant population. However, due to these variable outcomes, TMS for treatment-resistant depression is sometimes described as a lucky dip. So while it can be life-changing for some individuals, others receive no benefit. And one basis of this heterogeneity may be clinical or demographic factors, although these tend to show weak and inconsistent effects across studies. Another potential factor is suboptimal spatial targeting of the dorsolateral prefrontal cortex. However, over the past 2 decades, the optimal TMS target has remained elusive. So conventional targeting methodologies lead to quite variable stimulation targets across the spatial extent of the DLPFC. The first figure depicts 2 common targeting techniques, known as the 5 centimeter and F3 beam methods, which you are likely familiar with. These data are derived from 2 previous studies, and each dot represents the stimulation site for 1 patient. It is clear that there is substantial variation in the stimulation sites, both within and between these 2 methods, as well as more broadly across different targeting techniques that have been implemented in previous studies. However, this variation is completely reasonable given that the optimal DLPFC target site remained unknown. Another key point is that each of these conventional methodologies utilize a one-site-fits-all targeting approach. This obviously does or cannot account for individual specific variation in anatomical and functional neurobiology. As we learn more about brain connectivity and brain networks, it has become evident that this spatial scatter may actually result in different subsets of brain networks being preferentially targeted across different patients and targeting techniques. So various targeting methodologies have been explored, and to cut a long story short, connectivity-based targeting strategies appear to show the most robust relation to TMS therapeutic outcomes. The key question is, which brain connections should we be targeting? So mounting research suggests that TMS treatment response relates closely to connectivity with the subgenual cingulate cortex at the DLPFC stimulation site. And the initial reason for focusing on the SGC, subgenual cingulate cortex, is that this region was consistently implicated in antidepressant response across a range of different therapies. And on this basis, the SGC was also selected as the target region for deep brain stimulation. In the context of TMS, we're talking about functional connectivity, and as depicted in the middle panel, in this case, it is a synchronized pattern of activity between the DLPFC and SGC. So when activity in one region goes up, activity in the other region tends to go down, and this is known as anti-correlated functional connectivity, and this can be measured using MRI. So this relation between functional connectivity and TMS response is illustrated on the right in a cohort, across a cohort of individuals with depression, with functional connectivity between the DLPFC and SGC on the x-axis and clinical improvement on the y-axis. And each dot represents one individual. So moving left along the x-axis, we can see that stronger negative DLPFC to SGC functional connectivity at the stimulation site is associated with better treatment outcome. And this finding has now been replicated internationally across a number of independent cohorts. So for example, comparing the left and right panels, you can see that individual number 2, who had stronger anti-correlated functional connectivity at their stimulation target, depicted in blue, had a better treatment outcome compared to individual So on this basis, it's been proposed to use a one-size-fits-all group target. However, a potential disadvantage of a one-size-fits-all target is that emerging research suggested substantial inter-individual variation in network architecture, particularly across the spatial extent of the prefrontal cortex, illustrated in yellow and orange. And this higher diversity corresponds to increased evolutionary development of the prefrontal cortex. These areas can vary to such an extent that individuals can be reliably identified from a large sample based on their functional connectivity fingerprint. A few studies that delivered TMS to a singular one-size-fits-all group target using neuronavigation haven't really shown any compelling improvement in treatment outcomes relative to conventional targeting. And so together these findings provide a strong rationale for personalization of the DLPFC target site and defining the site according to functional connectivity with the subgenual cingulate. But in order to translate this approach clinically, we first need to know whether precise personalized targeting is even feasible. So to address this question, previous work by Ning and colleagues examined whether it is possible to reliably compute personalized functional connectivity-based DLPFC targets. And if this is possible, then the targets should be near identical when computation is repeated across two separate resting state MRI scans for a given individual. They tested this across 100 individuals and then computed the TMS target using conventional computational methodology. And in short, at best, personalized targets differed by 3.5 to 4 centimeters between scans, even when using the best quality scans from the Human Connectome projects. In other words, personalized targets were not reproducible and personalization did not appear to be feasible. Other remaining critical questions also include whether personalization is even worthwhile. Is there really much spatial scatter in optimal target sites across individuals, or are people quite similar in the context of subgenual cingulate cortex functional connectivity and DLPFC? Also, do personalized targets actually relate to clinical outcomes? So we embarked on establishing whether we can improve on this computational methodology to make personalization feasible. And once we had established this capacity, we examined whether personalized targets actually bear any relation to clinical outcome. So I'm going to present a brief overview of the results, and you can check out the papers for further detail if you like. So in short, we tested and developed new functional connectivity-based personalization methodologies. We then tested these out in 1,000 individuals with repeat scans from the Human Connectome project. We found that using this methodology, we were able to reliably identify personalized connectivity-based targets, and these varied by only 2 millimeters or a single brain voxel between scans, repeated when these scans were repeated the next day or even 1 year apart. Critically, we show here that inter-individual spatial variation in coordinates was 7 times greater than intra-individual variation, demonstrating that there is substantial inter-individual variation in optimal target sites, and that our approach preserved inter-individual differences. We also have familial data from the Human Connectome project, and we found that targets were more similar between monozygotic twins compared to dizygotic twins, non-twin siblings, and unrelated individuals, suggesting that these targets are stable and under genetic control. Finally, we tested the clinical relevance in a sample of 26 individuals who had previously undergone 13 minutes of resting state MRI and received TMS for depression, and for whom the stimulation target and treatment outcome was known. We retrospectively computed the optimal personalized stimulation target from each scan. We anticipated that individuals whose actual stimulation target was by chance closer to their optimal stimulation target would have a better treatment outcome. Now here you can see the scatter of optimal personalized stimulation target sites across this cohort. And here you can see that we confirmed our hypothesis. This figure shows that individuals who serendipitously received TMS closer to their optimal stimulation site consistently had better treatment outcomes. Moreover, these findings have since been replicated by Michael Fox's group at Harvard. Importantly, this relation between the proximity to the actual target site and treatment response was specific to functional connectivity-based personalized targets. In contrast, there was no relation between treatment response and proximity between the actual stimulation target and any previously proposed one-site-fits-all target, even when this group target was based on subgenual singular functional connectivity. There is now preliminary prospective evidence emerging from clinical studies that connectivity-based personalized targeting may enhance clinical outcomes across different brain stimulation modalities. These preliminary studies are showing increases in clinical efficacy for both TMS and deep brain stimulation treatments when moving from a one-site-fits-all targeting approach towards personalized connectivity-based target site selection. These studies suggest that this approach may be clinically transformative, but prospective randomized clinical trials are still needed, and we are trying to get the funding for this purpose. In the months since we've published this method, there's been global interest in implementing this approach, and these are the sites that are starting to implement our methodology. We're now working on a toolbox for clinicians to simplify implementation of this approach, and we're extending this connectivity-based approach out to other disorders. We'll be happy to collaborate in implementing this. So many thanks to Andrew Zaleski and to Paul Fitzgerald.